All right, so good morning, everyone, or good afternoon, or good night, wherever you are. And thank you for joining us on this second Cancer IO Sebinar. Today we'll focus on innate immunity, as you can see from the slide. It's organized by Cancer IO. Just a few words, it's a big consortium focusing on immuno-oncology. It's based in Finland, and it has over 10 big pharma companies, 10 or over 10 smaller Finnish companies, many hospitals, patient organizations, and universities. And for more information, please check the website cancerio.org or send an email if you like further information to cancerio-office@helsinki.fi. Today, the seminar will focus on innate immunity, as I mentioned, and you see the program on your screen. So today we'll have an introduction by Maija Hollmén. Then we'll have a talk by Juho Jalkanen from Faron Pharmaceuticals on their exciting new drug. Faron and Maija are both partners in Cancer IO. Then we have Eric Vivier giving a talk. He's from Marseille in France on I guess NK cells mostly, but on innate immunity in cancer therapy. After that, we'll have a brief Q&A. So all the questions, if you have any questions, please write them in the Q&A, which you find on the bottom of your screen. And if there's time, we'll handle those, but we'll probably not be able to answer all the questions. And then afterwards, there will be a panel discussion focusing on how to bring your findings from the academia to the clinic, both on the hopefully both on the commercialization side, but also on the clinical trial side. And the last thing I want to say is about the credit. So for students who want to get credit for this, like the first seminar, you need to attend and then provide us with a half-page seminar, a summary, a half-page layman's summary, which can be in Finnish or in English. You can send it to cancerio-office@helsinki.fi. The email address is on the screen. And if you've attended 5 seminars, that's sufficient for 1 credit, so 1 ETSF. So without further ado, let's start with the program. So we start today with Maija Hollmén. She's a partner in Cancer IO, and she, after her PhD at the University of Turku, Maija did a postdoc at the ATH in Zurich, after which she returned to Turku for a brief postdoc. After that, in 2016, she established her own lab at the University of Turku, where she's still working today as a young professor of tumor immunology, group leader, and academic research fellow. Her research exploits a unique scavenger receptor, CLEVER-1, to alleviate tumor-related inflammation and develop CLEVER-1 as a companion therapeutic, diagnostic, and prognostic biomarker for cancer patients. She does much of her work in collaboration with Faron, who will talk after Maija on this topic. And today, Maija has kindly agreed to provide a general introduction for the session. So please, Maija, the floor is yours.
Thank you very much, Jeroen, for this nice introduction. Just a moment, I'll share my screen. Okay, good morning, everybody. So my name is Maija Hollmén, and my lab is interested in harnessing macrophage activity to support anti-tumor responses. And I'll just give you a brief introduction on why are innate immune cells important for anti-tumor responses. And I just have to mention that we are also a part of the InFlame's flagship research on immune events in different diseases and how to promote clinical responses and clinical drug development to support patient benefit. Oh, sorry. So as you currently know, a lot of effort has been put to supporting T-cell mediated anti-tumor responses. And there is vast interest towards different checkpoint inhibitor molecules on T-cells so that they could be inhibited by, for example, ipilimumab, pembrolizumab, and nivolumab to support T-cell activation. And while there has been tremendous benefit on these drugs in a minority of patients, still the majority of patients do not respond to these therapies. And recent evidence shows that there are several other checkpoint inhibitor receptors on T cells, and there is active research on developing drugs towards these also. But I want to emphasize, and I really think that the way that things are now going on the T cell activation side is that it's more of a mass suicide of the lemming-like type. And there has to be some other ways on how we could improve anti-tumor responses. So we and others have thought of different ways to harness the innate immune system for improving these T cell responses against tumors. The innate immune system is very fast responding to different pathogen stimuli and consists of epithelial barriers and different phagocytous cells, dendritic cells, complement, and NK cells. And if we look at the bigger picture of this, what I showed you previously, that the most of the efforts on inducing anti-tumor immune responses have been to T cells. But T cells are just a small part of this immune system, which consists of innate and adaptive immunity. And what is known about the innate immune system is that it responds very fast within hours on encounter of different pathogens. It's semi-specific, so it can act in a similar way to various pathogens, and its diversity is limited. But in the last 5 years, there has been evidence that even the innate immunity can have a memory response. So it's called trained immunity, which means that when a phagocyte has encountered a pattern-like molecular species and reacted to that, it can remember this and react a different way to the second stimuli it sees. What is also important in this system is that there is these antigen presenting cells, for example, these dendritic cells that bridge the innate and adaptive immune system together to support the activation of the adaptive immune system. And how this works is that you first need the first signal, which is an MHC class antigen presentation to stimulate the T cell receptor and activate the first signal. Then you need a second signal, which is the CD28 from the T cell that binds to CD80 and CD86 on the antigen presenting cell. And this is the signal number 2. And here, for example, CTLA-4 can compete in binding to the CD80 and CD86 to dampen T cell responses. One important factor is that you also need a third signal, which is the cytokine secreted from the antigen presenting cell that then regulates how the T cell actually is differentiating. Is it differentiating to an effector cell or a T regulatory T cell, and so on. What is also important to remember in innate immune cells, that they have a very important effector function which regulate antibody-dependent cell cytotoxicity, antibody-dependent cellular phagocytosis, and complement-dependent cytotoxicity, which are very important in anti-tumor responses. Here you can see that the antibody that has bound to the target cell, which in this case is a cancer cell, this antibody Fc domain can bind these Fc gamma receptors and induce effector T cell effector functions towards these target cells. Raphael Kleins in 2000 showed that actually if you inhibit this or genetically deplete these Fc gamma receptor activating receptors on these effector cells, that you prevent totally the activity of Herceptin, which is a commonly used antibody drug to treat HER2 positive breast cancer. So if the main action of Herceptin is actually mediated by this antibody-dependent cellular cytotoxicity rather than the mechanism of action of its binding to the ERB2 molecule on the cancer cell. I think you have seen this image previously, but I just want to elucidate the importance of the innate immune system in regulating how the adaptive immune system is activated towards cancer. So this is a tumor where you have dying cells and they release danger-associated molecular patterns, which are then activating the innate immune cells such as macrophages and dendritic cells. these phagocytosing cells take up these antigens that are released from the tumor, and they traffic it within the antigens to the lymph node, where they encounter T cells, which then recognize these antigen presenting molecules on the dendritic cells and get activated. These T cells then start proliferating and traffic within the blood vessels to the site where these antigens are present, and then they can go and cytotoxically kill these cancer cells. And this cancer immunity cycle is very important mechanism how tumors are killed. However, there are many reasons why this doesn't happen. And this is just to show you that there is a very important interplay with different cells of the innate and adaptive immune system to control tumor growth. And for anti-tumor responses to occur, you need very different cell subsets such as mature dendritic cells, a Th1 response, these M1 so-called macrophages and NK cells, of course. Whereas normally what happens in tumor is that you get this tumor-induced immunosuppression, where you have a lot of these myeloid-derived suppressor cells, Tregs, M2 macrophages, and a Th2 response. And when this happens, you get escape of tumor cells and metastases, and ultimately the patient is killed by the tumor. So the idea would be that to support this, we need to kind of promote anti-tumor responses in different cells rather than just one cell. But I also want to emphasize that the more you push the immune system towards this anti-tumor immunity, it pushes back. So our immune system is in a performance so that it can, the more you push it, the more it pushes back. So you need to have a good control on the equilibrium of both of these situations. And lastly, I just want to show you one of my real stories on how smart these cancer cells can be when they regulate macrophage behavior. So I was wondering at that time when I was doing my postdoctoral research that why is triple negative cancer filled with these M2 type macrophages compared to the less aggressive estrogen receptor positive and HER2 positive cancers. So what I did was that I investigated in core culture system how these different cancer cell types were regulating macrophage behavior. And what I showed was that in the estrogen receptor positive cancers, these macrophages were really nice and these M1-like type, but when they were cultured with these triple negative breast cancer cells, they became these M2-like macrophages depicted here. So they were these alternatively activated ones that in Clever-1, and we'll hear about Clever-1 in just a minute or so. And this was fascinating to me, how these cancer cells were able to regulate macrophages to support their growth. And the identified factor that was regulating this effect was granulocyte colony stimulating factor that was highly secreted by these triple negative breast cancer cells. So I want to thank you for listening to this brief introduction, and I'll give the microphone to the next speakers. Thank you very much for listening.
All right, then I'll be putting my slides up.
Yeah, I will give a brief introduction.
Okay.
So thank you, Maija, for your contribution, and you will hear more from you during the panel discussion. So next is Juho, who was very eager to start already, so that's really good. So Juho works at Faron Pharmaceuticals. He has a master's degree in economics and business administration, and he has a medical doctor's degree from the University of Turku. And he's a fully licensed general practitioner and specialist in vascular surgery. He is currently a chief development officer at Faron Pharmaceuticals and involved in the development of this drug that Maija talked about. So today Juho will speak about his drug bexmarilimab, a macrophage switching drug targeting Clever-1 that is currently in clinical trials. So Juho, please go ahead.
Thank you, Jeroen, and hello, everybody. So as said, run the title of Chief Development Officer. Short disclaimer, although being an MD, PhD, I'm not an oncologist. I'm a vascular surgeon by training. Slides are rolling, I guess. Just making sure.
Yes, they are.
Thank you. So just shortly about Faron, we have currently 3 clinical development programs, one targeting Clever-1, which we're going to discuss about, then also inducing CD73 for organ protection, and then also inhibiting AOC3, also known as BAP1 for hematopoietic stem cell expansion. All of these come from over 30 years of world-leading immunology, and actually are original discoveries from Finland. Shortly, the pipeline the company is working on. So the big value driver is the program with Clever-1 and the IO stuff. But a lot to do for small Finnish biotech. Zooming in on the development program on bexmarilimab, that's the anti-Clever humanized antibody. Currently running is the first in human phase 1/2 study, which is now expanding to gastric cancer, melanoma, and cholangiocarcinoma. Then also a neoadjuvant study in a first-line setting in combination with a PD-1 in lung cancer. But let's move into the program and what does the drug actually do. So to understand the big picture of IO and where do we fit in, I'm kind of going with what Maija did, is that when anti-PD-1s, anti-CTLA4s came, they were miracles because suddenly they could cure metastatic melanoma and lung cancer, which were previously uncurable diseases. Although not a lot of patients respond, they were able to lift that Kaplan-Meier survival curve I'm showing on the left. But the sad part is that a lot of patients overall do not respond. And what do these patients that do not respond look like? Their tumor, the tumor mass can be, absolutely filled with them. And let's go back to the basics. Me being a surgeon, I need to also have a simple approach. The purpose of the macrophage is to digest debris and clean away. So in a big growing tumor mass, there's a lot of debris to be cleaned up. The thing with the M2 macrophages is usually what they clean, they're going to hide away from the immune system, not present to the immune system. So here in the immunohistochemistry stainings on the right, just showing some usually non-responsive cancers to checkpoint inhibition, for example, colorectal cancer, and then a melanoma patient from our own study showing that they're very high on M2 macrophages. That's the classical CD163 marker. And a lot of these M2s are Clever positive macrophages. And then basically you have no CD8 cells in the tumor. So no matter how much you're going to activate the CD8 cells, they will not work in this environment. Something will have to be done about this environment. So that's why we're targeting Clever-1. Clever-1, as said, is a big scavenger receptor. It's not expressed on all M2s, but a subpopulation of M2s, which we feel is the most immunosuppressive population of M2s. The thing with that Clever positive macrophage is whatever it digests, it doesn't present. So it will hide the tumor. It supports that immunosuppressive microenvironment by secreting IL-10 and activating Tregs. But when we inhibit Clever-1 with the humanized antibody, now called bexmarilimab, instead of hiding the tumor, the macrophage starts presenting the tumor. Instead of secreting IL-10, it starts secreting IL-12, TNF-alpha, and so forth. It starts activating the T cells. So I will show in actual human data that through targeting macrophages, we will get T cell activation and remove T cell exhaustion. Just first signs that this is a significant target and actually something that could, again, lift that survival curve up. This is from the TCGA database. Depending on if you're Clever high expressing, that's the red line, or Clever low expressing, that's the blue line, it actually has a rather significant impact on survival in all cancer patients, and then especially in patients treated with immunotherapy. And immunotherapy treated patients, it actually seems to dictate, do you respond or not? But it's rather logical if you think of it. Highlighting just the preclinical stuff in one slide. On the left-hand side, you have your classical clever knockout mice. So these are mice that are now deprived of the clever positive macrophage, but they have M1 macrophages. You try to plant a tumor into them. You try to plant lung cancer, lymphoma, or breast cancer into these mice that do not have clever positive macrophages. Tumors are rejected because tumors cannot hide anymore. And we get similar results treating PD-1 refractory models like Lewis lung cancer and B16 melanoma with an anti-clever antibody. We have single agent activity in these refractory mice models. And bear this in mind, because I'm going to show you this in man again. We seem to work where checkpoint inhibition does not. You again see the clever knockout mice and the immune cell infiltration compared to the wild type. Then on the right-hand side, next to the mouse, is an actual treated cancer patients. Pre-treatment, biopsy image, and then post-treatment. So we actually seem to get, even though now we're again doing anti-clever-1 treatment, we see an infiltration of cytotoxic granzyme positive CD8 cells into the tumor. What we see in analyzing these CD8 cells, that they're both interferon gamma and IL-2 secreting CD8 cells, so highly active and cytotoxic CD8 cells. What we saw initially in our just finding the dose, we could ignite around 30% of highly immunosuppressed cancer patients. What we know from our data is actually the colder the tumor is, the better the drug is going to do. And first clinical results already in dose finding. Here you see MSI negative colorectal cancer, which is known to be refractory to PD-1s, and another melanoma patient below that actually had received even an EP-Nivo combination, and that didn't work for the patient. But then again, anti-clever-1 as a single agent, after everything has been tried on the market, which didn't work, and then anti-clever-1 works. How is this possible? The thing here is that on the left-hand side, we show when we start treating, we neutralize clever-1, then it slowly comes back to baseline levels. As long as we got clever-1 inhibited out of the game, we will control also your usual checkpoints on T cells. Nicely coincides with the time scale of, as if we got our target covered, we will have T cell exhaustion removed. We're showing here only PD-1, but this similar curves are seen with CTL4, lag-free, and so forth. So it's controlling the T cells by targeting macrophages. Something completely new. Here again is showing a bit more. We got now already 140 patients. We see good clinical benefit in gastric cancer, cholangiocarcinoma, and then checkpoint refractory melanoma, as we did in mice, the P16 model. We see that approximately 30% of these patients benefit from the drug. The safety profile of the drug looks extremely good for an immune activating agent, because as said, if you push it too hard, you're going to get autoimmunity and a lot of bad stuff. But this seems to be very well tolerated. So first we did all comers in our dose finding study, and now we're already seeing signs in these 3 cancer types, which are now expanding further. Patients that seem to benefit from bexmarilimab treatment is their survival. So as we started the presentation looking at the anti-PD-1 Kaplan-Meier, the first thing to improve survival, and here we go, a macrophage target. with single agent activity that seems to be capable of improving survival further. Possibly this will be the next big breakthrough. This is just a summary slide to wrap that up. I've been working in the macrophage field for the past couple of years now, and I always hear there's no single agent activity with macrophage targets and you only need to combo and all that, but this sure seems promising compared to what I've seen in the field. Thank you.
Okay, thank you very much, Juho. And so remember, if you have any questions, please type them in the Q&A, and we can answer them at the Q&A session later on. So then we'll move on to the invited speaker and the main speaker today, and that's Eric Vivier. Eric Vivier is professor of immunology at Aix-Marseille University and at the Public Hospital of Marseille. He was appointed in 2018 as scientific director at Innate Pharma, a biotechnology company dedicated to improving cancer treatment with innovative therapeutic antibodies that exploit the immune system. He did his postdoc at Harvard Medical School and then joined Aix-Marseille University as professor at the Centre d'Immunologie de Marseille-Luminy in 1993 before becoming its director from 2008 to 2017. He's also one of the founders of the Marseille Immunopole, an immunology cluster created in 2014, linking fundamental and therapeutic research, innovation, and industrial development on the Aix-Marseille metropole. Eric Vivier's work focuses on innate immunity, and in particular NK and other innate lymphocyte cells. He has published over 380 scientific articles and is selected for the 2019 highly cited researchers from the Web of Science group. He is a laureate of the ERC Council. He had an ERC Advanced Grants. He's a member of EMBO, the Académie Nationale de Médecine, and the Institut Universitaire de France. Professor Vivier has received several awards, including those from the Ligue Nationale contre le Cancer and the European Federation of Immunological Societies. So we really look forward. So my apologies for my rusty French. It's high school level, so it's been a while. But we look forward to hearing all about the innate immunity and its role in cancer, and hopefully later a lively discussion about how to transfer your findings from bench to bedside and how you have experienced those steps. So thank you for joining us once more, Eric, and the floor is yours.
Well, thank you very much for the invitation. Thank you also very much for the very kind introduction. I will try to review for you today different topics. The main topic will be on NK cell engagers, and we will start by recapitulating why it's so important to harness cells of the innate immune system in the context of cancer therapy. So Maija Hollmén said it all already, so maybe I will go fast on these slides, and by the end I reserve for you, I kept for you some surprise, and but most of what I will be presenting today is actually unpublished. Okay, so this is my disclosure slide, and as you will see from the data that I will present, they are either part of the outcome of Innate Pharma or from my lab at the Center for Immunology at Marseille-Luminy at the Marseille Public Hospitals. We are actually trying to create bridges between academic research and the biopharma industry. So again, the menu for today will start by a small revisitation, so to speak, of what Maija has presented. She focused on where she presented the cancer immunity cycle pioneered by Dan Chen and Ira Mellman. And so we have recently proposed a new version of this, highlighting the steps at which the innate immune system was so important. So I will update you on what we call now the cancer innate immunity cycle. Then I will go on and update you also on what we've been doing in trying to harness the function of NK cell by using synthetic biology. And this is the NK cell engagers that are now becoming platform, which is called Innate for antibody-based NK cell engager therapeutics. And I keep the surprise for the end. Oops. Okay, so as Maija nicely said, I mean, it's obvious now that T cells can control tumors. And this has led to a very T cell centric view of the immuno-oncology world. But decades of immunology have taught us that indeed T cells are not autonomous in what they do. And they need the cells around them, such as the cells of the innate immune system, to become real strong effector cells and then memory cells that can protect against tumor development. So this is what I was alluding to. Back 2 years ago in Nature, we have actually proposed a review on the beauty of harnessing the cells of the innate immune system in the context of the new wave of immunotherapy. Because as we all know, immunotherapy has been wonderful over the last decade, but there are still too many patients who are resistant, either primary or secondary resistance to these treatments. And this includes not only immune checkpoint inhibitors, but also CAR T cells. So one expects actually to get a second wave of immunotherapy. And within this second wave of immunotherapy, targeting or harnessing the cells of the innate immunity makes a lot of sense. And here we have been highlighting, as I told you, with these kind of circles, the steps at which the cells of the innate immune system are so important in the ignition of protective T cell response. So within this, targeting NK cells makes a lot of sense. And this is a review that we recently wrote with Tom Gajewski, trying to highlight the benefits of harnessing NK cell function in the context of cancer. The first one is obviously to increase the capacity of NK cells to kill the tumor cell. This is why NK cells are known. But in fact, this is very important, but NK cells are also doing something else. They can produce cytokines and chemokines at the tumor bed and maybe elsewhere as well that contributes to the shaping of a protective immune response that will culminate into a T cell anti-tumor response. And NK cells do this by a number of mechanisms. And here we've been highlighting just two of them. The first one being the secretion of chemokines and cytokines, which are important for the priming of CDC1, which are by themselves extremely key also to the cross-priming of tumor antigen to T cells. So the CDC1 here can be activated by NK cells through the secretion of XCL1, CCL5, or FLAT3 ligand. At the same time, or in parallel, NK cells can also produce gamma interferon, and we know that gamma interferon can be extremely important also in shaping both the innate and the adaptive part of the immune system. So having said that, now we're going to turn to antibodies, because most of what we're doing, I should say all of what we're doing at Innate Pharma, is actually based on these magnificent medicines, which are the antibodies. So antibodies come in two flavors, as you know, blocking antibodies, as exemplified by the anti-PD-1, anti-PD-L1, but I will tell you a little bit about the new immune checkpoint inhibitor candidate that we call monalizumab, which is actually blocking the inhibitory molecule called NKG2A. And this is on the left side on this slide, but on the right side, we all know that there are also cytotoxic antibodies. And these cytotoxic antibodies, some of them have been clinically approved and participated really to the revolution of cancer treatment. And this is the case for rituximab and later on obinutuzumab targeting CD20. But we have also EGFR antibodies, such as cetuximab, which has proven efficacy in some conditions, and obviously trastuzumab, anti-HER2 in breast cancer in particular. So these antibodies are very different from the other ones. So the one in green, the cytotoxic, are really different from the ones in blue, which are non-cytolytic and just blocking, because of the FC portion of these antibodies, which is either silent by different means for the blue ones, or very active and even enhanced in its affinity to FC receptors for the green ones. Because these cytotoxic antibodies, they need FCR, FC gamma R, so cells expressing the receptor for IgGs, and these cells belong to the myeloid lineage, and also they include NK cells. And when NK cells do this, meaning when they recognize antibody-coated target cells, they affect what we call antibody-dependent cell cytotoxicity, or ADCC. So starting from this very simple scheme, when you have a cancer cell which is coated with antibodies and you have an FCR positive cells, what can we do to actually boost the function of the cytotoxic antibody? This is a very simple question that we ask. And one way of proposing some answers to this question was actually just to combine the two kinds of antibodies that I told you about. And this is what we have been doing, and this was published already 3 years ago when we developed an antibody called monalizumab. So now this antibody is in a phase 3 clinical trial in head and neck cancer, and I will show you why, by AstraZeneca. But this antibody was developed at Innate Pharma for its capacity to block the interaction between NKG2A, an inhibitory receptor expressed on NK cells and CD8 T cell subset. And NKG2A interacts with a non-classical MHC class 1 molecule called HLA-E. And as you can see from here, monalizumab is actually blocking this interaction. So why is it important? Well, because NKG2A is a prototypical example of an ITIM-bearing molecule. So ITIM standing for immunoreceptor tyrosine-based inhibition motif that we discovered years ago, co-discovered, I should say. And so when NKG2A is engaging HLA-E, then there is transduction through the protein tyrosine kinase phosphatase SHP1. It's producing an inhibitory signal within the cell that expresses it, namely NK cells and subset of CD8 T cells. So here on NK cells, it was very obvious to consider that if one can activate NK cells by antibody-coated target cells, and this is in green, maybe we can do even better than that, better than ADCC, by actually combining the treatment of the NK on the tumor cells, not only with an antibody that blocks the inhibitory interaction. And hence, the combination of monalizumab with one of the cytotoxic antibodies, and in our case, we focused on cetuximab, which is the antibody that recognizes EGFR that we know can be upregulated in several cancer conditions. And this is what we have been doing, so I'm not going over the preclinical data that have been published already, as I told you 3 years ago, but I will update you on the clinical results. And this is a positive phase 2 clinical trial that has been performed in a cohort of head and neck cancer patients. And you can appreciate, looking at this waterfall plot, that there is indeed an advantage for most of the patients to be treated by the combination of monalizumab and cetuximab. And as I told you now, this treatment is tested in a phase 3 clinical trial held by AstraZeneca. So this is basically the picture. I just told you about the left part of it, but I also, meaning that one can combine cetuximab, for example, and monalizumab to boost the DCC exerted by NK cells. But on the right-hand side, you also see that NKG2A can be expressed on subset of T cells, as I told you. And to be more specific, we could show, and this has been confirmed by others, that actually NKG2A is mostly expressed on PD-1 plus CD8 T cells. So it makes a lot of sense actually also to combine these two kinds of blocking antibody, the anti-PD-1, anti-PD-L1, with monalizumab, because if one wants to totally unleash the T cells, it makes a lot of sense actually to block these two breaks that exert inhibitory function on T cells, such as PD-1 and NKG2A. So this is also tested in clinical trials as I speak. But now we'll have to switch gears and tell you what we published 1 year later on the generation of NK cell engagers. So here we are not harnessing NK cells through the modulation of the inhibitory molecules that they express. We rather focus on the activating receptors. And you will see that we are using this synthetic biology to actually make a crosstalk between pathways which are very potent pathways expressed by NK cells. So this is what the NK cell engager version 1.0 look like, and I will update you obviously as the version 2.0. So here we are talking about a trifunctional natural killer cell engager that you see. So in yellow, you have a FAB fragment that recognizes the tumor antigen. In red, you just have a FC gamma 1, human gamma 1 portion that will interact with CD16, the activating FC receptor expressed on NK cells. And then in green, you have a FAB that recognizes NKp46. So basically, we are bringing together and bridging as well NK cells on one side here on your left and tumor cells on the other side here on your right. That's one. But we're not only doing that, we are also engaging the NK cell in a very active process, because we are cross-linking at the same time CD16 and NKp46. And there are reasons to do that. And the first reason and why we focused on NKp46 instead of just generating a bispecific antibody that would, or an ADCC enhanced antibody that will recognize the tumor antigen on one side and trigger CD16 on the other side. So the reason why we wanted to complete with NKp46 is that in contrast to CD16, NKp46 is expressed by all mature NK cells, as one can see here from the red graphs. And it's also specific of NK cells. It's not totally specific of NK cells because there are also ILCs, so innate lymphoid cell subset that can express NKp46. But by far, this is the best NK cell specific marker described at present. The second reason why we use NKp46 is that NKp46 is coupled to very potent activation pathways, because it's physically coupled in the membrane with high-time-bearing polypeptides, such as CD3 zeta and FcR gamma in humans, such as CD16. And as you can see, the engagement of both NKp46 and CD16, thanks to this non-covalent coupling with high-time-bearing polypeptides, initiate a transduction pathway that involves ZAP70 and or SYK and also PI3 kinase. So we have a full-blown activation of NK cells that culminate into cytotoxicity and cytokine and chemokine secretion. I put here on the left-hand side NKG2D because NKG2D is obviously a very interesting activating receptor that can be expressed also on NK cells and CD8 T cells. Problem is that in humans, NKG2D is exclusively associated with DAP10, and DAP10 doesn't initiate a full-blown protein tyrosine kinase activation. It does activate the PI3 kinase, which is very potent, but not the ZAP70 SYK pathway. So that's why we choose NKp46. And the last reason, this is very important in the context of cancer, is that NKp46 is quite stable in terms of surface expression in cancer conditions, which is not the case for CD16, for example, that you can see here being down-regulated as the surface of NK cells at the tumor bed in blue in the middle panel. And it's even worse with other NK cell activating receptor, such as NKp30. So for three reasons, we choose NKp46. Stable expression, exclusively, almost exclusively expressed on NK cells, so targeted treatment, and also a strong activation pathway linked to it. And the results, again, have been published 2 years ago. And here I just extracted 2 slides of this paper just to show you that in preclinical model in the mouse, here we are using a SCID mouse that we can inject with a human tumor cell expressing CD20, so it's a RAG-B cell line. And we've been generating here an NK cell engager that recognizes CD20 as a tumor antigen. And you can appreciate in red that all the mice were actually cured, which is very different from the absence of treatment in gray, obviously, but also more potent than what obinutuzumab, a clinically approved antibody, can confer. Interestingly and mechanistically, this NK cell engager activity is associated with the infiltration of NKp46 plus cells at the tumor bed, as you can see here on the right-hand side. So that was the first generation of NK cell engagers, the trifunctional. Now, I will update you on unpublished data showing that one can actually switch from NKCE3 to NKCE4. So what we added, you can look at the jigsaw puzzle, we added actually a fourth part on these NK cell engagers, which is a moiety, which will activate NK cells quite nicely. And this moiety is just IL-2. But we all know that IL-2 can be very toxic. And we also know that IL-2 can trigger Tregs. So we generated a variant of IL-2 that we call IL-2V, which will not bind CD25, the high affinity receptor for IL-2. So it's a non-alpha or alpha-less IL-2 that we generated. And the reason why we choose IL-2 is obvious. We know from the pioneering work from Steve Rosenberg back in the 80s at the NIH that IL-2 can be very powerful in treating cancer, but as I said, it can be toxic. So that's why we deleted the alpha chain receptor binding part. And second, NK cells constitutively express the IL-2R beta and IL-2R gamma. So there is no problem actually to trigger NK cells using IL-2 with an alpha-less IL-2 variant. And the idea here was actually to complete the strong activation pathways that are initiated by the engagement of both NKp46 and CD16 in our NK cell engager with the transduction pathway coupled to the engagement of the IL-2 receptors, namely JAK and STAT5 to induce strong NK cell proliferation. So this was a goal. And so to show you data now, so you can appreciate in red on the left-hand side that indeed the IL-2 variant that we generated doesn't bind CD25, in contrast to the IL-2 wild type in black. On the right-hand side, you can appreciate that it still binds this IL-2 variant, IL-2R beta very nicely, which is obviously key. So now we've been looking at the phospho-STAT5 as a marker of NK cell activation and other lymphocyte activation, STAT5 being a marker of the IL-2 pathway. So you can appreciate that when we compare just the IL-2 variant coupled to an anti-tumor antigen moiety to the IL-2 wild type in the same configuration, we still activate NK cells quite nicely using this different form of IL-2, but we lose a lot of activity on T cells, which is exactly the point. So you need actually 3 logs more of IL-2 variant to have the same activation of T cells. So it was exactly the case, what we wanted to specifically enhance NK cell activation over the activation of other lymphocytes. And we improve this when we actually couple the IL-2 variant within the frame of these NK cell engagers, because as you can see here, now when we have the tetrafunctional, tetravalent NK cell engager, we can readdress the IL-2 variant directly to T cells, because this molecule will have 4 heads. So besides the tumor antigen and the IL-2 variant, it will have binding parts of FAB, recognizing NKp46, and also the FC that will interact with CD16. And I think that this slide is self-explanatory. So now what's going on in terms of activation, really, when we want to dissect the role of each part of this tetrafunctional molecule? Well, you can see that in green, you have the wild type tetrafunctional molecule with the IL-2 variant, the tumor antigen binding part, the NKp46 binding part, and the FC. And it works quite nicely. Now, when you lose the FC binding part with an FC silent in light blue, or when you lose the NKp46 binding part, which is in dark blue, or when you lose both, you can see that at each time you lose efficacy as assessed by the EC50, you lose 1 log at each time almost. So you need everything in the single molecule, and I will show you this using other readouts as well. So as expected, the NKCE4 is actually inducing a nice proliferation of NK cells, as you can see, actually much better than a targeted IL-2 variant, which makes a lot of sense mechanistically, obviously. And the first generation of NKCE, the NKCE3, doesn't induce any proliferation of NK cells. Now, in terms of cytokine and chemokine secretion, same story. You can appreciate that the full tetrafunctional molecule is inducing a very nice activation of NK cells, and you need each part to have the optimal activity. I would like you to pay attention on the last curve in green open circle, when you see that in the absence of a tumor antigen targeting, so when we delete the FAB that recognize the tumor antigen, it's always CD20 in this set of preclinical data. You can appreciate that we do not induce cytokine or chemokine secretion at these doses, showing that there is no off-target activity of the NKCE4, which is obviously extremely important. Now, in terms of lysis, same story. We have very nice lysis using the NKCE4, and we lose activity as we lose the CD16 or the NKp46 binding possibilities, and again, no off-target effect as well. We've been challenging this data obtained with primary human NK cells, now going to the mouse, and you probably know that ADCC is quite inefficient using mouse NK cells. And we know some of the reasons for that, but you can see that using obinutuzumab. You see that obinutuzumab is inducing some ADCC. We are using RAG again as the tumor cell target, but it's not much. This can be improved using the first generation of NK cell engagers in light green, but look what we have in the bright green when we have the tetrafunctional molecule is much better. So what about in vivo now? But in vivo, I've been working on NK cells as it was said in the introduction, in the kind introduction, since Harvard back like 20 plus years ago, and I never saw something like this. Here, by just 2 treatments with the NKCE4, you can totally abolish the proliferation of the tumors in the mice, as you can see in green, and you can appreciate that one needs again the whole part of the NKCEs of the tetrafunctional NKCEs to have this activity. We've been comparing here with a single injection, the activity of NKCE4 with obinutuzumab, and I think that it reads by itself. The NKCE4 are much more efficient. Now, mechanistically, this is associated with the tumor infiltration by NKp46 cells that also express granzyme B. So NKCE4 induce in the accumulation of activated NK cells at the tumor bed. We confirm these results using qPCR, and this is not only activated NK cells in terms of granzyme B expression, but also in terms of gamma interferon secretion. Now, we've been dissecting the mechanism behind this. This is obviously NK cell dependent. When you deplete NK cells, you don't see the control of the tumors no more. And by dissecting again the parts of the molecule which are important, you can appreciate that you really need the 4 parts of the NKCE4 to have this activity. I would like you to pay attention on the differences between the green part and the red part. So the green part is showing you the tremendous activity of the NKCE4. The red part on the far right is telling you that this is achieved only when you have the 4 parts of the molecule in a single molecule. Why? Because here we've been treating the mice using a NKCE3 with a tumor antigen binding part, NKp46 binding part, and C16 binding part, and IL-2, but in a separate molecule. You can appreciate that this has some activity, no worries, when you compare the red part on the far right, to the to the black graphs on the far left. You can see that there is indeed an activity, but much less than with the tetrafunctional molecule in green. So we've been challenging the activity of this NKCE4 using a disseminated model, so-called metastatic model using the melanoma B16F10. And you can appreciate that the activity again is just huge, tremendous activity of the NKCE4 where you don't see any proliferation or almost no proliferation of the tumor cells in the lung of the injected mice. So obviously the problem was toxicity. So we went to the monkeys using a pre-tox model in cynomolgus. And as you can see here, by targeting again CD20, one induces in the monkey a very nice depletion of B cells. So if you look at the right part of the graph in green, at 0.5 mg per kg, one sees a very nice depletion of the B cells in the monkey, which is already apparent at 0.05 mg per kg. Interestingly, this is not associated with any signs of toxicity. This is very important, obviously. One can look here at the clinical data, behavioral, body weight, rectal temperature, heart rate, oxygen saturation, name it. We don't see any differences between vehicle-treated monkeys as compared to NKCE4-treated animals. And now we've been looking also at biological parameters. So in the serum of these treated monkeys, one can see upon 6 hours of injection, an increase in interferon gamma, IL-6, MCP1 and IL-10, but I would like you to pay attention on the axis, on the scale of the y-axis, because you can appreciate this is not much. We are in the scale below nanograms and much below nanograms, and for interferon gamma and IL-6 in particular, and IL-10 as well. And look at what we have. What was published, I should say, using a T-cell engager targeting CD20 in the same monkeys, same species of monkeys, and so T-cell engager targeting CD20. You can appreciate that the production of the pro-inflammatory cytokine is actually 100 times higher. So there is obviously an advantage in terms of toxicity to harness NK cells as compared to T cells. So basically, to wrap up what I've been telling you so far, the NK cell engager technology provides efficient engagement of NK cells against tumors. We've been targeting NKp46 for the reason that I mentioned. There is co-engagement of CD16 and NKp46 for the NKCE3, and co-engagement of CD16, NKp46, and the IL-2 receptor, IL-2R beta, IL-2R gamma for the NKCE4, and this leads to unprecedented control of tumors in preclinical models by NK cells. So now we call this platform Enquête for antibody-based NK cell engager therapeutics, and here I want to thank all the people, all my colleagues at Innate Pharma who have been participating to this. Now, if I have some time, do I have some time left? Because I didn't check the time.
Yeah, you still have at least 10 minutes left.
Oh, wonderful. So I would like to switch gears and tell you more about what we are doing on the other side of the campus, because Innate Pharma is on the Center for Immunology on Marcel Lumini campus. We're on the same campus, which is obviously very important for this bridge between basic research and biopharma. And I want to tell you more about other cells which look like NK cells, but which are not NK cells. So we've been reviewing the field of innate lymphoid cells already some years ago, as you can see here from this paper. And in particular, I would like to tell you about NK cells and ILC1s. You know that ILCs, innate lymphoid cells, are coming in different flavors, but ILC1s are very close to NK cells by a number of parameters. First of all, they both produce gamma interferon. And then also in terms of phenotype, both in human and mice, they can look pretty similar depending on the numbers of molecules that you look at. But in particular, in the mouse, when you look at lineage negative or CD3 negative, B220 negative, but NK1.1 plus and KP46 plus cells, you will have both NK and ILC1s. So we wanted to know more about two things that I will present to you today. First, what are the developmental differences or similarities between NK and ILC1s? That's one. And second, can we use state-of-the-art technologies to distinguish more carefully between NK and ILC1s? So we've been focusing on mouse liver for the first part for one reason. As you can see in the panel A, one can discriminate quite nicely between NK and ILC1 in the mouse liver. This is probably the only organ in which it's so clear with so few markers. So here again, we select lineage negative cells. we select NKp46 plus NK 1.1 plus cells, and you look at CD49A, CD49B, and one can see with CD49A ILC1s and CD49B, one can see NK cells. So we've been looking at other markers, and as you can see here, there are other molecules that can participate to the distinction between ILC1 and conventional NK cells, here is referred to as C NK cells. In particular, CD200R1 is expressed on ILC1 and not on NK cells. Same thing for 12, same thing for CD69. And actually on CD69, it's important to mention that a major difference, biological difference between ILCs and NK cells, and ILC1 are no exception to that, is the fact that NK cells can recirculate and one can find them in the blood or in the lymph, which is not the case for ILCs in general. Innate lymphoid cells are tissue-resident cells, and ILC1s, again, are no exception to that. Now, in terms of transcription factors, both ILC1 and NK cells express T-BET, which is seen here, which is obviously in relation to their capacity to produce gamma interferon. But ILC1 do not express EOMs or EOMS, in contrast to NK cells. Now, in terms of functionality, as I told you, both can produce gamma, but NK cells are better at producing gamma. This is the other amount for TNF. And also NK cells are cytolytic using degranulation and perforin, which is not the case for ILC1s. So the first observation came from our colleagues in China, Hui Peng and Xingang Tian at Hefei University. And basically, it was very puzzling. They could show actually that when you look at liver ILC1, they cannot be reconstituted by bone marrow transplantation. This is very weird because we know that most of lymphoid cells can be, and NK cells in particular. But here, when you compare the efficacy at the NK here, the ILC1, liver ILC1 reconstitution upon fetal liver transplantation, FLT, versus bone marrow transplantation, BMT, you can appreciate that it works very nicely with fetal liver, but not with bone marrow. So to make a long story short, and I have no time to show you this, but we could show this is also the case using adult liver cells and not only fetal liver cells. And one could identify in the adult liver a population of cells that we call LSM for lineage negative SCA1 plus MAC1 plus that you can see on the upper graph here. There are not many cells, but they are there. And one can find them in the fetal liver as well, but one do not find them in the bone marrow or in peripheral blood. And when you reconstitute irradiated mice, here it's a congenic model using these LSM cells, one can see that one can reconstitute quite nicely and specifically liver ILC1s and not NK cells. So it was very puzzling to us. And so we've been trying to dissect what was going on here using single cell RNA-seq when we actually identify a different subset that can reconstitute these liver ILC1s. So again, I have no time to go into the details, but basically LSM cells are very heterogeneous. They come in different flavors, as you can see here, with 12 clusters. But we could actually restrict the capacity of this LSM cell subset to produce liver ILC1 by focusing on these cells which are lineage negative, CD122 positive, IL-2R beta, and CD49A positive that we call cluster 5. And you can see here that this cluster 5 actually, and cluster 3, cluster 5, but cluster 5 is probably the most important here, are really the cells that look like ILC1 and can reconstitute liver ILC1 specifically. That was the first part. So there are populations in the adult liver that are included in this LSM population and also included in this population of CD122 plus, CD49A plus that can give rise selectively to liver ILC1. What are the mechanisms? So we and our colleagues could show that it was totally gamma interferon dependent, which is a surprise because the role of gamma interferon in hematopoiesis is not as clear as the role of gamma interferon in the effector function within the immune response. But here you can see that looking at mice that lack interferon gamma or mice that lack the interferon gamma 1 receptor, the receptor 1 for gamma, there is actually a quite important decrease of liver ISC1, and it was specific of this population of cells. Now, when one injects interferon gamma by using a plasmid hydrodynamic injection, one can really nicely reconstitute liver ISC1. So there is an interferon gamma dependent pathway. But very surprisingly, this is not dependent upon lymphocytes, at least adaptive lymphocytes. You get rid of CD4 T cells, you get rid of CD8 T cells, you get rid of B cells, and nothing happens. So now we took advantage of a mouse that we have produced years ago, which is an NKp46 Cree mouse, in which we can target the genes that we want under the restriction of the NKp46 promoter region. And thanks to this model, we could actually cross it to interferon gamma flux mice. And as you can see, this is a very nice model in panel A, where you can see that NKp46 cells and only these NKp46 cells do not produce interferon gamma no more, which is not the case for NKT cells, for example, which can produce interferon gamma. I see a production induced by PMA ionomycin, but we have the same result using IL-2, IL-12, IL-18 stimulation. So here we can ablate the capacity of NKp46 positive cells to produce gamma. And what you can see in the panel C is that actually we can reproduce the decrease in liver ISC1, specifically when we ablate the capacity of ISC1s, of NKp46 to produce gamma. We previously described, as seen in this slide here, that when we got rid of NK cells selectively and we knocked out AOMS from NKp46 cells to do that, one can see that again, so the absence of NK cells has absolutely no impact on ILC1s, because you see that in those mice, you lose NK cells, but you don't lose ILC1. So basically, what we are showing here is that gamma interferon is important for the normal development of liver ILC1s. That these liver ILC1s come from progenitors which are present in the liver. So the in situ differentiation of liver ILC1s, which again is gamma interferon dependent. But the cells that are producing gamma interferon have to be the ILC1s themselves, because this is not T cells, and this is not either the NK cells, because again, when you get rid of NK cells, you don't have any impact of ILC1s. So basically what we are proposing here, and this has been published earlier this year, is that there is a new pathway during which, or in which I should say, liver ILC1s develop locally via an interferon gamma dependent pathway. You have LSM cells here in the adult liver that can differentiate into liver ILC1. And this is the interferon gamma produced by ILC1s and not by NK cells, which can actually harness the activity of LSM cells because they express the interferon gamma receptors, which is not the case for the other cells. So we do think this is quite interesting because for the time being, the generation of lymphoid cells were restricted to bone marrow progenitors. This is not the case here. So maybe the tissue resident ILCs, besides ILC1, are produced during embryonic life by embryonic progenitors that could seed different organs, such as the liver, and then they develop in situ and they self-renew according to environmental cues. And this is what we are seeing in the liver. Now to finish, and this is very last slides, as I told you, the second part of the new data, totally unpublished, that I wanted to share with you, resides in our aim to better differentiate NK and ILC1 across organs. And so we have used CytSeq to do this. We have been sequencing using CytSeq and Technique Genomics more than 50,000 individual cells coming from liver, spleen, salivary gland, and gut. And as you can see, there is a tremendous heterogeneity of this NKp46 plus NK1.1 plus. One can think that looking at the facts, that this is a homogeneous population. It's not at all an homogeneous population, as one can see here. And I have no time to go to the detail, but we could actually identify different subsets of NK and ILC1s, as you can see, and different signatures within these different organs. And to make a long story short, what we could do is actually to classify this different subset into 3 subsets of NK cells, NK1, NK2, NK3, and 2 subsets of ILCs, ILC1s, ILC1A, ILC1Bs, an intermediate subset, which are differently represented in different organs. Okay, and final slide. I forgot about this one, sorry, but I'm happy to take any questions you would have on COVID-19. This is the story that we published last year, actually, when we tried to repurpose molecule that we had in hands at Innate Pharma in the context of severe COVID-19 pneumonia. And we've been able to identify the complement pathway, in particular the C5a pathway, to be important in the inflammation that leads to severe COVID-19 pneumonia and put patients in danger. Okay, I will stop here. I already thanked all the people at Innate Pharma who collaborate, who are working on NK cell engagers. And I want to thank people at the Center for Immunology who participated to the NK versus ILC1 story. And I will stop here. Thank you for your attention.
So thank you, Eric. This was a really excellent talk with a lot of different aspects of NK cell biology and actually ILC, which I had never heard of. There are a few questions, but maybe I start off. But because I'm as I said, I don't know these ILC. So what do these cells do in biology? What do they do in the body, in healthy or in tumor case?
Okay, so ILCs are resident lymphocytes, as I said. They belong to the innate immune system, and they don't express any TCR or BCR. And they have a propensity to be present in all inflammatory sites. We see them as amplifiers of the immune response. The first line of sensing would be epithelial cells or myeloid cells. And thanks to cell-cell interaction and or cytokine secretion produced by these epithelial cells, and I'm thinking here a bit at IL-33 in particular, but could be also IL-25, these so-called alarmins, but it could be also myeloid cells. There are signals which are integrated and detected by ILCs. And then ILCs, which can be very plastic by themselves, can secrete an array of cytokines. So ILC1s would do gamma, ILC2s would produce so-called Th2 cytokines, such as IL-4, IL-13 in particular, and ILC3s would produce IL-22. And so they participate to the regulation of the immune response. And we could show already 3 years ago in collaboration with Alain Fischer that selective deficiency of ILCs do not have any dramatic consequences in terms of protective immunity, at least in the context of modern medicine and modern hygiene, but they are probably very important in the control of inflammation. To tell you in a nutshell what ILCs are about, but please read our review in Cell, which is not that old, and you have more insight, you will have more insights on what they are doing.
Okay, thank you very much. So we have a question here by Fiona Simpson Fraser. So she asks, which NK subset has the most antibody-mediated anti-tumor activity?
Well, so definitely the CD16 positive cells, because to recognize antibody target cells, you need the Fc receptor expressed on NK cells. That for sure. Now there are different subsets of NK cells which have been identified very recently, such as the adaptive NK cells, which only have CD3 zeta as a transducing molecule associated with CD16. So as a consequence of that, as zeta has 3 items and comes as a homodimer, there is 6 items per CD16 molecule, which is better than when you have FcR gamma, which has only 1 item. So this subset could be the one which is the most important in vivo, but we don't know that. But definitely CD16 positive cells.
Okay. All right, and then Matti Kohonen asks, he says, great talk. So you use a subcutaneous Raji cell model, and while other groups use a systemic IV model. So what are the differences and or advantages of using this subcutaneous model?
Well, those are different questions. We are using the subcutaneous model to mimic solid tumors. Obviously, there are limitations to this model, but this is commonly used. When you inject the cells IV, we are rather mimicking a disseminated tumor, such as metastatic tumors. So those are different conditions. And if you pay attention to what I said, we've been using two models, the subcutaneous model, but also the IV model, but we use B16F10 because it's a more classical model for disseminated tumors.
All right. So I had one question about this, the first part of your talk about the monalizumab, because you say you use it with cetuximab and that's now in phase 3. But then you also have, as you mentioned, there's also a reason to combine it with PD-L1. So have you identified biomarkers, or do you pre-select for biomarkers which treatment arm to do?
Yeah, this is an excellent question. We are all trying to do that in the field of IO. This is in progress, and I cannot disclose what we have right now. We are working on it.
Yeah, but it makes sense, right? Too, because you need to choose which one to use, and I'm sure you will find something there. And then to come back to those ILC cells, so because you say that they kind of start, or they kind of produce themselves, or they help produce themselves, right? They secrete interferon gamma, and then that leads them to production of more. But so how do the first ones come there? Is there any evidence or theories about that? Because there needs to be a first one to start the interferon gamma.
Yeah. Well, we don't know that. We don't know what are the environment, the liver cues, which selectively trigger the interferon gamma production by ILC1s. We just don't know. And we don't know the pathways that are prior to this gamma amplification to the seeding and the development of the first ILC1 progenitors. There are much more to understand and to dissect, for sure.
Okay. So then one question from Sarah Olila. She had a general question on ILCs. Are there established roles of ILC2/3s in anti-tumor immunity? Do they in general expand by proliferation upon inflammation?
Yeah, okay. So there are many reviews and many articles. Actually, unfortunately, maybe more reviews than original articles, and apologies because we might have contributed to this, trying to understand what is the role of ILCs in cancer. There is a concept, which is a concept of tumor infiltrating ILCs. There are papers also showing that ILCs can participate to tumor immunity. In collaboration with Gabriel Belz, we have a paper which just came out in Nature Immunology showing that indeed, in the context of melanoma, ILC2 can be very important for anti-tumor immunity and can be unleashed using anti-PD-1. So there are also other studies that I have no time to summarize here on the role of ILC3 in tumor immunity, in the lung cancer in particular, because ILC3 has the propensity to help in the formation of tertiary lymphoid organs. So it's not a direct activity against the tumor, but it can participate to the anti-tumor immunity. And for NK versus ILC1s, NK have recognized anti-tumor function, but ILC1s can actually be pro-tumoral. We, in collaboration with Mark Smith, we had another paper in Nature Immunology years ago showing that indeed there is a propensity for some subset of NK cells to become ILC1s, upon TGF-beta in particular treatment. And once these ILC1 are at the tumor bed, they might be detrimental for the host. And we have actually unpublished data right now on this model as well, confirming that ILC1s can be bad guys in the context of cancer. Just in a nutshell, to show you the complexity and how much we need to, how much more we need to understand.
Okay, so then there's one final question for Eric, and I will then ask still one question to Juho. So Rosa Stahlberg would like to know from Eric, do you use some matrix to establish your tumor models in vivo?
Sure, absolutely. What we usually do, so in the subcutaneous model, what we do is that we inject a specific number of cells, obviously the same for all the mice treated in the same conditions, and then we let them grow until a specific threshold, which can vary from a mouse model to another one. And once we are at this stage of growth of the tumors, then we randomize the mice and treat them using different kind of treatments as we would do in a randomized clinical trial. And then we assess the growth of the tumor sub-Q by direct measurement of the tumor growth. Okay.
And so then one question to Juho, because there were no time for questions. Because this comes from me, so I think you showed this really fantastic survival curve for responders and non-responders. But how do you define responders in that case?
It's the RECIST criteria for disease control. So the PR or SD in a follow-up CT image.
Okay, so if there is some kind of control in the tumor, so it doesn't need to go all the way, but if there's some kind of control of the tumor growth, then the patients survive all of them.
Yes,
but they don't not
so far.
Yeah, so far, sure. All right, good. So thanks everyone for the questions and everyone.
I ask a question, Jeroen. Sorry to interrupt. I know we have not much, but I had a question for Juho. Very nice talk, by the way. Congratulations on that.
Thank you.
There's something I didn't get. The antibody that you have against Clever-1, is it a depleting antibody or just a blocking antibody?
Well, it's not depleting. We don't want to do not deplete them, reprogram them. We actually call it a functional antibody. It changes the function of the macrophage or monocyte it targets.
But so what is the isotype? It's an Fc silent antibody?
No, it's an IgG4, but Fc is silent, yes.
Yeah, but you know that IgG4 can bind very nicely to CD64.
Yep.
Okay, so it's not Fc silent. But also in one of your slides, you had S Clever-1. Do you mean that you are blocking the soluble form of Clever-1?
Yes, the science is a bit more complicated, but due to the sake of time. So the Clever-positive macrophages also secrete soluble Clever-1, and soluble Clever-1 itself can bind directly to CD8, CD4 cells and inactivate them. So we need to neutralize also the soluble form.
Okay. Do you know the receptor for soluble Clever-1?
Yes, but it's we're not giving that away yet.
Okay. Thank you.
All right, excellent. So thank you, Eric, for some additional questions. So now we move into the panel discussion part, and this we'd like to focus because at Cancer.io we are at the interface between academia and companies, and more and more focus nowadays in academic science and also by the funders is really the applicability of the science that you're doing and also the potential to commercialize it. And every country has big programs on the way to get scientists to really think about commercialization. So we feel that this is an important discussion to have, and that's why I'm very happy that Harriet Gullstén will join us today. So she is in Business Finland. She's a senior advisor at Business Finland since 2017. She main know-how is on health and well-being industry, company coaching, innovation funding, internationalization, and ecosystem building. And she's worked before Business Finland in many international companies, but has also done research at universities. So she's familiar with both ends of the spectrum. And she has a deep expertise in genetics, molecular biology, microbiology, immunology, pharmacogenetics, and diagnostics. And at Business Finland, she's been, for example, involved in the FinGen project, but also in preparing personalized health program. She's been facilitating companies to go to the market and internationalize with using Explorer funding and market access programs in Asia. And she really, her main passion and goal is to create growth, success stories, and diversity. So, and as I mentioned, she represents Business Finland. So Harriet, how does Business Finland support early stage pharma biotech companies, especially drug development companies? And are there any other funders in Finland, and how do they collaborate with Business Finland?
Okay, thanks for the invitation and nice introduction. I'm happy to be here. So we do fund Finnish companies in preclinical and clinical phases, mainly bio loans, but also with the grants in a preclinical phase. And we also found phase 3, but the company needs to have the product, the drug, which they will bring to the market themselves. So IPR and know-how selling is not the goal, and according to our alliance, what we really can fund. And who else can fund? We have ELY centers, TESI, Finnvera, FinFund, and some cities has their own funding as well available.
Okay, and does Business Finland collaborate or coordinate with those funders to work together on programs?
In programs, we are creating Team Finland. So we have certain type of the collaboration, but and we have the same CRM system, but certainly we know each other and collaborate. Yes.
Okay, yes. So thank you very much for this introduction to Business Finland. So Juho, so you work at Faron, and Faron has been established a while ago, I guess. But so how did Faron get things off the ground in Finland, and have there been any special opportunities, but also challenges in general, and also maybe in Finland?
Well, yes, sure. Getting off the ground is relatively easy in Finland if you have that commercial mindset as a scientist, because preclinical work can be shifted into a company rather easily, and funds for that are available in Finland to a certain extent. But it becomes difficult when you move into clinical trials, which are very expensive and cannot be solely done in Finland, for example. So then I would say, even though Business Finland have great motivation to try to move there, but there isn't substantial funds to do that and to support clinical trials in Finland, and you will have to start looking abroad very early. Already when you're setting up preclinical, you will just start to have to go out and become international. There is no existing funds in Finland to support clinical development.
So Eric, so you're a co-founder of the company. So how was that in France? Any differences? Are there any major funders for this kind of for the commercialization? And is there a lot of for example, venture capitalist funding available?
Yes, so it's very difficult to compare with something I don't know. I just heard about what's going on in Finland like 5 minutes ago, so I will not compare. I can just tell you what can happen in France. Obviously, it's a new story at each time, but we started this company back 22 years ago. We have been helped at first by the French government, but then we went directly to venture capitalists. And then we decided to go with deals. So we had the first deal with actually a Scandinavian company, Novo Nordisk, that you probably know. Then we went through a deal with BMS, and then another one with AstraZeneca, and then another one with Sanofi. So these deals help you raising funds and also progressing in the clinical development of the molecule that you are developing. And in this path, we've been also listed at Euronext first, and then at NASDAQ back 2 years ago. And so this is all I can tell. So again, I think that there's no magic recipe. If there is one, please let me know. And I think that it's really dependent upon a lot of factors. Some are intrinsic to the company, which is fair enough, but some are unfortunately very much extrinsic, like like COVID-19, for example. What can happen, which can happen and really disrupt entirely your business for different reasons, or promote it. That depends what you are doing. If you are promoting efficient anti-COVID-19 vaccine, then it's good news for you. No, that's what I can share. There are different ways of trying to translate results of basic research into new drugs for the patients. That's all.
And so for Innate Pharma, so when did you make these agreements with the big pharma? At what stage? Before clinical trials, or did you first have your first clinical trials under Innate Pharma and then you made the deals? This may be different per drug.
Well, that depending upon the molecule. I mean, we have quite a rich portfolio of molecules. So the deals are not for the entire portfolio. Obviously, they are non-exclusive agreement or exclusive agreement molecule per molecule. And depending of the molecule, sometimes it was before, sometimes it was after the first phases of clinical trials.
Okay, so it just depends on how things go and how the data.
Absolutely, and what is the appetite for the molecule that you have, what is also the expertise of the partner. So there are very different factors.
All right. And so because of course for academic researchers there's two ways to go. Either you have your research and you commercialize that into an own company, but the other option is to work together with pharma companies and to then kind of not co-develop but collaborate with them and then see your findings move forward. So but then I think a lot of, I mean, my experience talking to pure academic researchers is that they're all afraid of the pharma, or afraid is a big word, but they are afraid that the working together with the pharma will result in delayed publication and also limits to academic freedom. But Maija, you work quite closely with Faron, or maybe very closely. So have you experienced any of that? So what is your opinion here?
I think it very much depends on the company which you work or whom you work with. So I can give you 3 examples. During my PhD studies, I was working with Genentech. They had finished or quit this program investigating ERP4 antibodies. And I got through an MTA, I got 32 antibodies, which I was trying to discover a splice isoform specific one. And I found one, and I did a manuscript on that and sent it to Genentech. And after 1 month, they returned it and said, yes, you can publish this. And in the meanwhile, they had patented all the findings based on the publication. So that was really fast and it was really efficient. And I can just say good things about our collaboration with Genentech. Then the second example is Novartis. So during my postdoctoral search, I was interviewed by Novartis presidential postdoctoral fellowships. And I went through the company there and interviewed a lot of people. And they said that I could only work on known things like PI3 kinase pathway or something that they was just investigating for some purposes, but it wasn't really fascinating science or anything like that, because they didn't want to publish anything related to their new findings. And though, of course, I didn't go there because I wanted to do good science. And the third example is Faron, which is, of course, a spin-off company from the University of Turku researchers. And there, I think the idea is more that we want to publish everything that we find out. Of course, we do the patenting before we publish. But I think new knowledge is a key point for starting companies and their products to be like, or increase awareness of these scientific findings and interest towards the company and gaining money to go clinical trials.
Okay, so that's yeah. So that sounds a bit mixed experience, and because we work in our lab also with a few and we have had very good experience with the pharma companies we work with, and they are very collaborative and so on. Of course, you need to sign MTAs, which makes a lot of sense, right? But other than that, it's been very nice. So maybe a question to Harriet at this stage. So because maybe a lot of researchers are now thinking that I have this finding in my lab, and then I but I don't know, is this now fundable? Is this commercializable? Is this interesting? I mean, scientific, it's nice, but so from my own experience, when we set up Cancer.io, there's been a lot of interaction with Business Finland, and they've been really helping us out. So what would you recommend to scientists? Obviously, this is again dependent on the finding and stuff, but what would you recommend scientists to do when they're in doubt or when they think about these kind of things? When should they contact Business Finland, and how?
Yeah, good question. We are here for you. So basically, when you have an idea on your mind, please contact us. If you don't know any particular person, so just go to our website and send the either call or send the message. But basically, yes, we are helping out, and of course nowadays we don't fund that much research as in old days, but still we have co-creation and research, the old total funding research to business funding, and I have been evaluating those projects for 2 years, and there has been some drug development projects. So keep in mind that maybe your project can be, if it's from the research organization, it can be also research to benefit R2B funding, and we are funding it with the 20 million euros annually. So we have 2 calls on a spring and fall time.
Yeah. And from our lab's experience, this tutorial, or now it's business to know, what is it? Research to business. Mm-hmm. Helsinki University has the Helsinki Innovation Services, and there's a call, I think, once or twice a year, and they help you really prep your case for Business Finland. So they help you out in to focus your talk and your what is it, your pitch, and all of that. So I don't know if the other Finnish universities have similar, but I guess they also have some similar.
It depends on the university, but in many universities we have this pitching session, which the university or innovation unit they organize, and they are asking our help to sort of come and comment the projects, and they will give open comments, and it's just before research teams are sending the application, so it can be very useful to join those sessions.
All right, good. So we got now some information about commercialization, and maybe also a little bit about how to do that in practice, at least in Finland. But I also wanted to very briefly talk about clinical trials, because this is something that we see, I think, because again, I'm speaking here from my own point of view, and also maybe about from some others I talked to, because you do great research in your lab and in the academia, but then to bring this to the clinic, that's really, of course, next level. And from own experience now, it's a lot of work, and there's a lot of things to consider. So Juho, maybe starting with you. So Faron has started now a clinical trial, which I think runs in Helsinki and in other places in Finland, but it's also running abroad. So can you tell me a little bit about the process, and did you find any help for this or in Finland or abroad, and what are the processes here?
So good thing in Finland, especially in the field of oncology, our clinicians are science orientated, and they are used to running clinical trials. So actually you can get rather well started in Finland, but we're such a small population that you can't really go much beyond phase 1 in Finland. So you'll need international sites, preferably big sites that can enroll, a lot of cancer patients. And for that, it's actually just hard work, going out there, talking your science, meeting a lot of people. The thing you will need is to have compelling science. The clinical trial landscape. It's a marketplace, and you need to get your science out there. You need to get people excited about your science so that they would want to run a clinical trial with your drug, because the biggest and best clinical trial sites have a lot of competing trials ongoing, and you need space there. So getting those champions on board in different sites, for example, Gustave Roussy in Paris, which Eric knows well, they are important breakthroughs for your clinical development and important development partners. And especially the US side, it's even more competitive and more like a marketplace, but you'll need to go there for, if you're going to raise VC money, they want some US sites, US names behind you backing up your science. Have to say though, US sites, it's even more difficult getting them going from Finland, and per patient costs are many times higher than in Europe, and especially from Finland. So actually you can do relatively a cheap start in Finland with Finnish clinicians, but you will soon again have to go abroad, have to go present your science and get people excited about it.
All right, so we seem to have lost Eric already, so we thank him anyway. So because we've done a little bit of research on this, and indeed, as you said, internationalization is important for your clinical trials. And also, what we've understood is that especially Finland has a fairly isolated population for decades. So just doing your study in one country might also lead to genetic, that certain an outcome might not be representative for the whole world population, and that might be, for example, there was apparently some kind of vaccine some years ago that had been tested everywhere but in Finland, and then it came to Finland, and then some people got extremely sick because the Finns have some kind of weird mutation that it's not so relevant, but here it was really important.
Yes, you'll never get a drug approved doing only studies in Finland.
Well, that anyway, for sure, for phase 2 and 3, you need to go abroad anyway, but maybe the first steps can be done, and yes. Once you have set it up, then you can maybe expand.
And we have good oncologists here in Finland, so thank you for them.
Yeah. So what is the first step to take your drug? Is that fun? Well, you guys were a company, so maybe the funding was then in the company, but
it basically, well, depends when you come out of the university or the lab. When there's a big push also, what we haven't covered is actually having that drug manufactured, which already needs a lot of investment. There actually, Business Finland is very good, and they get the manufacturing part, and those funds are rather suitable, those loans, to get the manufacturing done, to take the drug into man. And that also needs to be, taken into consideration. So it's not just having something from the lab and starting a clinical trial. There's a lot more that goes with it and has to be considered. But there are, people who've done this in Finland, not that many, just going out and asking for advice also. I think we need to have that more in Finland, because we're not a big population. We need to really, support each other on what we do.
So Harriet, maybe you can chime in here. So as you already said, that there is some funding from Business Finland for drug production. So how does Business Finland support clinical trials?
Well, I just touched it in my earlier answer, but clinical trials we fund, but only for SME companies. Because we see that mid caps and big companies, the impact is not sufficient for them. So in that sense, it's clearly possible for phase 1 and 2 and in some cases 3, but very, very rare cases in phase 3.
And I'm guessing that as when you identify your own drug as an academic, you need to set up a company first to then do the clinical trials, because you don't fund academics for this, I guess.
Exactly. So it needs to be Finnish company or subsidiary with the OU.
Yeah, exactly. Yeah.
So basically companies and yeah, please. So
No,
so yeah. So basically, that any company who is buying, let's say, a preclinical or even phase 3, phase 2 phase drug or drug candidates, they can be funded with a loan, but those cases are not I mean, companies who buys drug candidates in a phase 2 level, they exist in it, and our alignments has done according to the current situation. It can change certainly, but at the moment it goes like this.
Okay, so because we are now on at the end of the session, so I think what we can conclude from the panel discussion is that there is good doctors in Finland and opportunities for funding. And I think what Juho said and what we've also found and what Harriet will probably underline very much is, you know, don't chew on this yourself. If you have good ideas, go out there and find networks, go to Business Finland, talk to your innovation service, talk to other scientists, right? Try to get advice about what to do next, because, you know, you cannot do this alone. No one can, and get the right people around you to do this. Okay, with that, I want to thank all the speakers today and Harriet for joining the panel. And I would like to thank also all the people. So we had at some point 150 people listening, which I think is really excellent. And so, yeah, and then thank you everyone for your participation. And we hope to see you next time at the 3rd Sebinar, which will be early autumn when we have planned that. Okay.